Good afternoon. I'm Jennifer Pietenpaul, the Chief Scientific and Strategy Officer for VUMC, and it's really my pleasure today to introduce our On the Horizon here in October, a leap forward for precision medicine, NashBio, and the Alliance for Genomic Discovery. And this is just a exciting area for discussion today in presentations. And I just want to point out the tremendous work and leadership that has gone in to having this moment of celebration. And that comes from our executive leadership of Nashville Biosciences, both Leeland Ekstrom, the CEO, and Elizabeth Ann Stringer, the Chief Scientific Officer, as well as our partners and collaborators in this Alliance for Genomic Discovery, and really have brought tremendous leadership, and that is Illumina through Will Ferguson, who's the Vice President for Pharmaceutical and Biotech Collaborations. It's because of this partnership between Nashville Biosciences and Illumina that we're able to actually highlight the major accomplishment that is the focus of the town hall today. And so, as I've just alluded to, we have several distinguished presenters today who are going to provide important information to the research enterprise and the academic enterprise, and that includes our guest, Will Ferguson, who's the Vice President at Illumina. From Nashville Biosciences, both the CEO, Leeland Ekstrom, and the CSO, Elizabeth Ann Stringer. And our very own from Vanderbilt University Medical Center, our Senior Vice President for Personalized Medicine, Dr. Dan Roden, who will present and offer some great musings on the science that we can all do from the great product of this partnership between Nashville Biosciences and Illumina. So our agenda today across our speakers is first, Leland's going to give us a history of Nashville Biosciences. I think this is very important. And then Will is going to tell us, how did this Alliance for Genomic Discovery come to be? And in reflecting back, I think Will and Leland, this is over 2 years of really hard work from both of your teams that have culminated in this alliance. And it's really exciting to think about. Elizabeth Ann Stringer is going to talk to us about what the alliance is. And then, as I mentioned previously, Dan Roden is going to talk about the science that will be enabled by this alliance. And then we have many distinguished faculty who are joining a Q&A session to answer your questions, and that will all be moderated by Travis Osterman. And so really looking forward to this town hall today, and I hope you are as well. So Leland, please take it away.
Thank you, Jennifer. Can everyone hear me?
Yep.
All right, take control. Technology works. So yeah, as Jennifer said, we're going to tell the Alliance story in 4 parts today. I'm going to start with the history of NashBio. I came to Vanderbilt almost 9 years ago now, summer of 2015, to help build what has become NashBio. And so I'll give the historical perspective and then hand it over to Will and Elizabeth Ann to talk about how it came to be and what actually it is. So in a lot of ways, NashBio is a continuation of work that you all have been working on for almost 20 years now at this point. The story of NashBio starts all the way back in 2004 when the story of BioView first got underway, and some of the folks on this call, as well as others who've been a part of it along the years, really just thinking about the foresight and the innovation that came together shortly after the Human Genome Project, and bringing the pieces together that ultimately became BioView. So the medical record and the DNA samples. And in a lot of ways, or in all ways, we couldn't do what we do on a daily basis, or certainly with the Alliance, without that foundation that you all had put in place over that period of time. I'm not going to go through BioView. I'm going to assume a fair bit of fluency with BioView and what it is at this point, other than just to highlight the research success stories that have come up over the years. When you look at the scope and scale of what BioView has grown to at this point, in terms of the number of users and the projects and the high profile publications that have really advanced our understanding of health and healthcare and how genetics drive that, as well as the resources that have come back to the medical center. I think it can only be characterized as a full-throated success on the research side. And per the prior slide, we were created to help extend that into the commercial space. How to move this forward. And I'm still struck at this point. Dan, I don't know if you remember this, but it was shortly after I came to Vanderbilt, you commenting about the number of companies that had reached out to you through the early years of BioView and asking how to make use of this type of resource in their own. R&D activities. So in thinking about what BioView can contribute to commercial R&D, there's a few things that are worth highlighting. The first is the value of human genetics, and I think the well-recognized understanding of human genetic evidence in successful pharmaceutical R&D. There's actually been attempts to quantify this over the years. Some papers I've got listed down in the footnotes, trying to measure how much more likely is a drug with human genetic evidence to make it through clinical trials. And early estimates said of twice as likely, kind of twofold improvement in probability of success from having that genetic evidence. More recent attempts have put it more like 6- and 7-fold likely to get through the pipeline. So just a real understanding and a recognition. The number of pharmaceutical companies have built up human genetics and genomics efforts based on the back of this, and certainly it's something that appeals to those companies. Second aspect is the ever-growing focus on real-world data, and it was the 21st Century Cures Act that catalyzed a lot of this. By no means the initiator of that. I think real-world data is a buzzword that goes back much earlier than that. But in terms of trying to push it into all different aspects of the healthcare ecosystem, whether it's the regulatory processes, and I think Pfizer managing to get a new indication using just real-world data a few years ago. I don't think there's been a ton of other examples since, but it's still a good example to highlight in that regard. But also then thinking about pharma and their expanding use of real-world data, and really just trying to understand how drugs and therapies are used in the clinic as opposed to in clinical trial. And then the third source of value is just the efficiency that you all built in terms of wiring this into the provision of care. So whether it's pulling off that medical record, the fact that the samples are all collected or kind of continually collected, and it was that prospective collection that was in a lot of ways a catalyst for the AGD, for the alliances, compared to some of the other competing initiatives that are out there where they're actually going to have to wait for samples to show up. But also just things like the longitudinality of the medical record and having all that history attached to patients. It allows for some really elegant and really intricate study designs that just wouldn't be possible without that history. So again, that kind of efficiency which unlocks all kinds of interesting scientific questions. However, certainly some challenges to overcome, and that's where the NashBio parts of the story came in. What we were designed to address some of those challenges, and I'll just highlight a few of them briefly. Certainly having a prospective sales effort, our business development and marketing team, and creating marketing awareness, but also building up use cases over time and figuring out how to promote those. Obviously, the papers I highlighted on the earlier slide are a big awareness grabber, but pivoting those into how can companies actually make use of these data and these data sets, and doing it in a way that really speaks to them. One of the other challenges that we all recognized in the early days was the difficulties in contracting, and particularly quickly. And I don't say this to talk down to anybody on the legal enterprise here at the medical center, but oftentimes when you have two big institutions, you have UMC on one side and a large pharmaceutical company on their side, the interactions between entities of that size just don't move very quickly. And so we were created as kind of the nimble intermediary, just to make it easier to do the types of things we're going to describe today. Third challenge with company academic interactions is getting expert resources, all of you and your postdocs and grad students, to do science that isn't necessarily their own. There may be overlap in topic and thematics, but it's still somebody else's science ultimately that you're doing from a commercial standpoint. And so building up resources that can do that science without having to convince people to do something they may not be wholly interested in. And then fourth challenge to overcome, and that's moving from a time and activity billing. Oftentimes when companies pay academic institutions, they very much expect to know tell me what it's going to cost, and that's exactly what I'll pay you, as opposed to what value you might be helping create for them. So kind of all across the board, different challenges that we recognized in the early days and sought to overcome with NashBio. Fifth one's a little bit different than some of these, and this was something that Gordon Bernard had pointed out in the early days of NashBio, is just with the wealth of information and science in BioView. There's so much science, but only so many scientists. A recognition that the number of people we have to do things here at the medical center is limited by, or limits the impact of BioView in some regard. And so the idea of extending it to the commercial space, really opening up its impact and trying to maximize the impact it can have on health and healthcare. So with that, we were created and we positioned ourselves as a genomic and clinical analytics company, and really that theme of accelerating or making it easy by trying to sand off all of those rough edges on the prior page, with a broad remit to work across the space. We didn't come in with any preconceived notions of this is what NashBio has to do. We were kind of guided to go think broadly and widely, and work across pharma, diagnostics, and life sciences partners, and find the most productive use cases for the resources we had. So we were launched March 2018, so coming up on our 6th anniversary early next year. And it's really that fusion of the data that comes from Vanderbilt and the BioView resource analytics capabilities we've built up, and then a customer service focus as well. And that's something we really do try and push ourselves on, is to make sure that whatever we're producing is useful to the companies we're working with, and not just throwing something over the fence because it's a deliverable in a contract. We worked with more than 75 companies over our history, and I'll throw this up to just give some examples of who those companies are over the years. The important point being, it's from the very smallest. So Goldfinch Bio was an early stage startup when we worked with them. They've actually gone through the whole life cycle at this point. They they closed the doors early last year because the specific question they were working on didn't pan out. But then large pharmaceutical companies, and everything in between, the companies we worked with. And then just to give you a sense of size and scope of NashBio at this point, we've grown to about $10 million in annual revenue. We have 25 people, give or take, some of them sitting here in Nashville, but a lot of them scattered across the country at this point. So what do we actually do on a daily basis? And I'll illustrate that with just some of those use cases and some vignettes from some of the companies we've worked with to accomplish them. The first one was working with a company called Genuity Science. In a lot of ways, this was a precursor to what became the Alliance, but using that very precise, or I guess very deep information in the medical record to build some very precise cohorts for ultimately further studies. So being able to develop things like progression cohorts and responder non-responder cohorts and very exquisite disease cohorts, and then simply generating the data as a starting point. So pulling out whole genome sequencing, and then positing it back in BioView, as well as enabling Genuity to pursue their subsequent studies. The next category, and certainly is linked very closely to the first category, and in a lot of cases we'll help, or we'll take on those analyses ourselves, whether it's using things like genome-wide association studies or GWAS to identify new drug targets, or using something like a PheWAS study to identify new disease or side effect indications. Example of this was a project we did with Takeda a couple of years ago, trying to understand why some patients progress on to more severe forms of the underlying disease, and other patients appear to get stuck or stay at a more stable point, and trying to understand if there's specific genetic markers that are driving that progression versus not. Third set of uses is what we call clinical trial optimization, and certainly with the identified nature of BioView, we don't run any clinical studies directly. We can't recontact the patients, but we can certainly help design protocols. We can certainly help understand where patients are sitting at a place like VOMC, and that was the example we did with Calcimedica a few years ago, was trying to understand the specific disease population that they were trying to find. They were having trouble recruiting patients for it, and so trying to profile where those patients move through a place like VOMC so they could speed up their recruitment. Synthetic control arms are also an example of the kind of work we do here. So basically taking a single arm clinical study and then applying the same criteria to extract the baseline standard of care to inform or put that single arm study in better context. And then the last example is things like cost effectiveness and comparative effectiveness studies, and we work in just the pure rural data side alone. Vital Transformation was a company we worked with a couple of years ago after the introduction of the first CAR T therapeutics, and trying to understand how those CAR Ts ultimately changed the cost of care and the burden of care for patients with acute ALL. I don't remember which of the exact flavors of it was, but a couple of the early, and one of the early other CAR T indications. So trying to understand how those patients respond from a healthcare utilization standpoint after they're on the drug. So with those as kind of the day-to-day activities, and this is where we'll pivot towards the Alliance. Our most ambitious project yet, I guess, is the best way to describe it. And it was an idea that was born, or that came about, it's actually about probably almost 4 years ago now, was the idea of moving the whole BioView infrastructure side to a much deeper and much bigger genomic data set. For those familiar with the details of BioView, certainly the 100,000 DNA samples that were genotyped in 2016, 2017, it was a tremendous effort and a tremendous resource for research. But in a lot of ways, genotyping has become old technology at that point. It's a couple generations old, and it wasn't taking full advantage of the entire samples. Likewise, kind of those project by project initiatives I gave on the prior side, or even academic labs, sequencing 1,000 here, 3,000 here, 5,000 here, was going to take a long time to really expand the resource to its fuller potential. And so we came up with the idea of trying to put a relationship together that would ultimately lead to whole genome sequencing the whole resource and sequence as many samples as we could. And that was the launch pad, or the jumping off point for the Alliance. So with that, I'll hand it over to Will and can take the next chapter.
Thank you, Leeland. Okay. All right. Is it still in full presentation mode? Okay, great.
Yep.
Okay. Well, I'm going to jump right in. So Illumina, just for those that aren't as familiar who we are, a global genomics company with its mission being to improve human health by unlocking the power of the genome. We are celebrating our 25th anniversary this year and have been a key contributor to driving down the cost of sequencing, key innovations in the field, as well as the genomics revolution in the last 2.5 decades since the sequencing and completion of the first human genome. Our sequencing and array platforms support R&D across vertical markets and applications, including oncology, genetic and infectious diseases, reproductive health, and beyond. Our technologies empower health care providers to leverage genomic insights in clinical care, providing patients with diagnoses and groundbreaking therapies and companion diagnostics. Our platforms have support over 400,000 publications with over 20,000 instruments installed globally. I'm going to say next slide, but I have control, so I'll do it. For the past 3 years, and as mentioned previously, we've been working together across VMC, NashBio, and Illumina to build a differentiated and highly valued R&D resource. These interactions were across the organizations at the highest levels and across departments, and takes advantage of the strong foundation that is BioView and what NashBio has continued to build upon. The breadth and depth of this resource has been acknowledged by a top KOL in the industry as spectacular. And with that, we at Illumina wanted to catalyze the generation of deep genotyping data in the form of whole genome sequence data that could be coupled to the deep longitudinal health record data in BioView, bringing together two of the best of breed and their respective areas of expertise, led us to forming the synergistic partnership between Illumina and NashBio, as well as bringing an unparalleled data resource for research to life. And although Illumina and NashBio had a great vision for this resource, we still needed more support and funding to really build and generate the whole genome sequence data for the cohort in its entirety, the full 250,000 samples from the biobank. And so that resulted in the formation and launching of the Alliance for Genomic Discovery, which was announced this summer. And prior to that, we actually kicked the project off earlier in the year with Decode Amgen in January. And this announcement of starting the project with the diversity ancestry cohort helped us to build enough momentum with as we continue to work with other industry partners, resulting in the full formation of AGD in July and this resulting announcement. These biopharma partners are supporting this alliance as they also see differentiated value in this resource. The depth of the clinical data, as Leeland mentioned, combined with whole genome sequence data, provides a robust path to breakthrough discoveries to advance research across disease areas. And so with that, I really want to kind of go through what we call the stories behind the story. And I'm not sure the animation. Okay, let's see here. Okay, more animation. Sorry about that. As our respective teams at Illumina and NashBio went through the various peaks and valleys that are inevitable while embarking on business development effort of this magnitude, these stories began to emerge at critical inflection points along the way. And the ones we're going to highlight today are Fortune, Fire, the Power of Deadlines, and Takeoff. And so I do want to mention some themes that really both Leeland and I and others across the teams would highlight, and that is really three things. First is perseverance, second is flexibility, and the third is creativity. And so with Fortune and Fire, and that was really the foundation of the partnership between Illumina and NashBio. We although we had really started to build some momentum and had gotten some great feedback from the market, we realized we needed to get that partnership in place. And JP Morgan Healthcare Conference every year is really a kind of a watershed conference. It kind of kicks off and sets the agenda for the year in this space, and was really is important to a lot of organizations in the life science tools and pharma industry. And so there was a significant amount of momentum and interest from the senior leadership within Illumina to go ahead and get this inked up before the end of the year. And Illumina, in fact, has a shutdown from around Christmas to New Year's. So we had limited time as the clock was ticking. We were all working feverishly across both sides to get this done. Unfortunately, we didn't actually get there. We fell a little short at the end of the year, a little disappointed and concerned. However, everyone rolled up their sleeves in that last week before the conference was to start. The first week of January, we were actually able to work through and get everything done just in time to make the announcement, hence the Forged in Fire title there. And so this is a really critical milestone for us to achieve in time to really, make it known that this is something that was happening. It was real, and really it turbocharged our business development efforts in the industry as we moved ahead. Fast forwarding past that inflection point, we moved to kind of the second, contract that we were pushing. Spent the year lobbying and building a lot of support from the prospective BioAlliance participants and partners. However, we really wanted to get something just started. And so this is really where we were able to take advantage again, JP Morgan, this time starting in 23, with our engagement with Decode Amgen. And we had started to engage them in the fall and really started to lay the groundwork for what's called the bridging agreement and getting that in place before JP Morgan. With 3 parties involved, the added complexity and again, importance of getting it done. We actually, this time around, went ahead and worked through the holidays. And this is, these are folks, legal, business, scientific folks across all organizations through Christmas and New Year's, and held multi-hour conference calls, almost daily through that holiday season when most people are enjoying their time. We were working through this, and we were able to get it done right up into to the very to almost hours before we had everything ready, and were able to execute it the night before and make the announcement that Monday morning, again, which was absolutely critical to advancing our efforts. And finally, moving into the next phase, we had spent following the success and the announcement and real foundation of interest and the alliance participants saw on the previous screen. We were in earnest negotiations through the spring and early summer. However, we still had to actually get the contract executed to kick off and announce the full formation of the Alliance for Genomic Discovery. And this was extremely complex, required the perseverance theme, and as well as creativity and flexibility along the way. And so we actually worked through several multi-hour sessions across legal teams for all the different alliance participants. And in fact, I had multiple versions of what we thought would be the final agreement that everyone would execute. It happened 3 times, so 3rd time's a charm. And we did work through a lot of things at the 11th hour that we thought were resolved. Nonetheless, the strong partnership that we had in place allowed us to just continue to work through and persevere. And in the end, the value, the resource itself, all of the incredible characteristics and attributes of the data that exist, and just really what allowed us to get us to to takeoff. And now the Alliance is live, and we're really excited, generating data by the week, and real excited about the next phase. Elizabeth Dan.
Elizabeth Dan, I think you might still be muted.
Yep, thank you. Can everyone see the slides?
Yeah.
Okay. I cannot see them.
Do you need one of us to advance for you there?
That sounds great.
Happy
Thank
to do so.
you.
Sure.
Thank you, Travis.
All right.
Okay. Great. Thanks, Travis. Appreciate it. So I'll be answering the question, what is AGD? So I think many of you know, in summary, AGD is an alliance between NashBio and Illumina and 5 pharma partners who are funding the whole genome sequencing of 250,000 DNA samples from BioView. Those 250 samples, we've broken them into 2 cohorts. The first cohort of roughly 35,000 individuals are those of high likelihood of African ancestries. And that cohort has actually completed sequence generation. Then we have the remaining 215,000 cohort, and those are selected by random sampling. And you can go to Starbright to learn more details on the sample selection. And then our pharma partners, so AbbVie, Amgen, AstraZeneca, Bayer, and Merck. And Travis, if you can advance one more, we have an announcement that it's no longer just 5 pharma partners. We now have 6 pharma partners. If we can go back. So BMS joined the alliance last week. It's really exciting and really a testament to the value of this resource for drug discovery. Thank you for the applause. An initiative like this, 250,000 samples don't get sequenced overnight. So it is a multi-year effort. We began that effort in January of this year and spent the first part of the year really ramping up the operations, making sure everything ran very smoothly, all the QC steps in place. And then in July, once we were fully ramped, we began our regular cadence of monthly shipments of around 13,000 DNA samples. For those of you who have been at VUMC for a while, you know, the old storage freezer and an RTS for selecting samples really could not have done something like AGD, had an initiative this large with a resource like that. So really appreciate VUMC and their investment in resources that made this possible. And then I think what's of main interest is the actual data itself that gets generated and released. So the whole genome sequence data is released on a quarterly basis. And most critical to the Vanderbilt investigator community are these 2 releases that I have highlighted in red with the stars. So that is the joint call for the VCFs, so the multi-sample VCF data. For cohort 1, that sequence data was generated and then processed, joint call performed and released in October. And then the joint call will happen again at the end of the sequencing effort, late March of 2025 for the full 250,000 cohort. So then moving forward, just want to briefly touch on the workflow here. Vantage is doing the plating of the samples and then shipping those to Iceland, where Decode is performing the sequence generation. Once those sequences have been generated, they then upload the data to Illumina's Illumina Connected Analytics platform, which serves as the data management and analysis platform for AGD and further processing of the data. Moving on to the next slide, I think it's really important to talk about scale here. And here I've made the comparison between the mega genotyping data set and our whole genome sequence data set. So the whole genome sequence data is roughly 225,000 times the size of the mega genotyping data for an individual. So if we think about 90,000 individuals with mega genotyping data, we could represent that with a loaf of bread. If we wanted to represent 90,000 individuals with whole genome sequences, that's going to be a blue whale, the largest animal on the planet. It's just remarkable the size difference we're speaking about here. And then Travis, if you can advance to the next slide. Of course, for AGD, we're not stopping at 90,000. We're talking about 250,000. So here, the entire data set for mega, again, represent that with a loaf of bread. AGD, the entire data set is going to be 2 blue whales and 4 orca. So just really, really different scales that we're talking about here. Travis, if you can move on to the next. So there's careful considerations that we need to make. So storage size and cost for data this large. And then, of course, the analyses we're going to be performing and compute and cost for those analyses on the data. And it's going to take a retooling. It's going to take different approaches to this data set. So this is not a data set where you are just going to download the entire data set to your hard drive and perform your analyses there. It is going to take cloud functioning and in a different way to think about these data. Okay, what's available? We'll stay there. The data package itself. So we have CRAM files that are being released. That allows us to back convert to the raw files. Important to keep those. And then we have our variant call files. Again, the most important, I think, to the investigator community are going to be those joint calls, those multi-sample VCF files. Cohort 1, again, already released, and then for the entire 250,000 at the end. And then for interpretation of the data, we have Nirvana's annotation library, which is a compendium of essentially any annotation tool or database that you can imagine is there and made available. The processing of the sequence data, so we're talking 30x coverage. Again, this is run on Illumina's Dragon pipeline, and the reference map is the GRCH37 reference map, which is what UK Biobank used. So good comparison there. And then Vanguard will be performing a comparative study on the GATK pipeline, which is used for all of us, and the Dragon pipeline on about 5,000 of these sequences. Travis, you can advance. Thank you. So data access. Again, the data are all being released on ICA. ICA is the management and analysis platform for AGD. It's built on top of AWS, and storage compute charges are at cost. The tool was designed for a broad spectrum of users. You have GUI, you have your notebooks. The top screenshot is of a notebook available in ICA, and then command line capabilities for those more comfortable with command line. ICA will have limited support from Victor and BioView for the VUMC community. So where should Vanderbilt investigators go should they want to access this data? So you are going to request access just like any other BioView existing genetic data project. So I've also included a screenshot there of what the Starbright page looks like. You'll easily be able to find where you can go and register for access to the data. For the data, workflows, and analyses, those are going to be available on Terra. And again, a screenshot of that at the bottom. The Victor team is in the process of bringing over those 35,000 MS VCF files. Once those have completed transfer, which we're hoping by the end of the week, that data will still need to be QC'd before they can be made available for broad use by the research community. So not there yet, but very, very close to making this data available. And then the last point to make is no restrictions or embargoes for VUMC investigators for using this data. There is one exception, and that's for industry-sponsored studies and sharing of data. And that's just for the first 3 years post-release of the data. And with that, I will turn it over to Dan.
Dan, I think you're muted.
I was asked to comment on, what could we do with a resource like this, and I feel very awkward because I feel sometimes like I'm just not creative enough, and there are many creative people on this call who will do things that I haven't thought of yet. But here are some ideas that I hope will happen. So if you cast your mind back before there was a Human Genome Project, the way to find new genes for diseases was to accumulate very large cohorts, very large kindreds of families with a disease. Some members had the disease and some members didn't. So traditional linkage analysis. Along comes resources like the one we're talking about here. So this is the famous Wellcome Trust analysis of 7 common diseases in 14,000 patients. So we thought that was a gigantic study in 2008. It's a tiny study right now. And they just showed that you could do genome-wide association and find loci across the genome that appeared to be associated with common diseases. That was a new way of discovering new genes. And then there is this idea that this slide has appeared in many incarnations, but this is the review article that actually first proposed this idea that there are, I don't know if I can use a pointer. Can I use a pointer? Who knows? Cursor. That in the lower right are most variants are identified by GWAS are common and have very modest effect sizes. On the other hand, variants identified not by GWAS but by family studies are in Mendelian disease. Those are very rare or rarer and have much higher effect sizes. So this is the sort of typical cartoon that many people have reproduced in many different ways. This is the original version. And I just highlight this idea of Mendelian disease, and that what they put on their slide in 2008 was penetrance. And I actually think that's probably the wrong thing. It's effect size, but penetrance is really sort of something completely different. I think the way we think about it now. So Elizabeth Ann showed this in terms of size of the resource. In other words, how much computer storage you need to store the data. But in fact, another way of looking at it is how many variants are we looking at? So in the mega data array that we have, there are around 1 million, somewhat north of 1 million SNPs that can be captured by array technologies. And what we have here, we think, is going to be more than 1 billion SNPs. I base that number on numbers that are coming out of all of us right now, and at least 10,000 structural variants. So it's not only a matter of scale in terms of how big the genomes are, but how much variation there is in those genomes. And The advantage that we have in our environment, I think, is that we have a very large EHR, which is a single EHR, and we have a large number of faculty members who understand how to mine it and how to make relationships between mined phenotypes and genetic variation, one at a time or multiple sets of variants. So this is my sort of shopping list, but I'm sure that there are other things. So probing extreme phenotypes. So why is it that there are people who have who are very tall or very short, or people who have extremes of body weight, or people who smoke heavily and should have lung cancer and live to be 98 years old? Those are extreme phenotypes, and we can find them in BioView, and now we can relate them to rare genetic variants, and that's a direct path to drug discovery, I think. So finding drug targets, one way is to do it with a phenotype-based approach. Another is to do it with a genotype-based approach. Here's a person with a rare genotype and an unusual phenotype. Is that a drug target? The granddaddy of that probably is PCSK9. And many of you know the story around PCSK9 and its discovery as a drug target, and then its rapid development as a drug target. I think personally that when we use terms like breast cancer or atrial fibrillation or heart failure, we're talking about multiple disease subtypes. Those fields recognize that there are multiple disease subtypes, but there are more disease subtypes to be defined. And it's only with a very large data set of genomic data and phenotypic data that you can do that. So rare Mendelian diseases, I think they're not so rare. In BioView right now, there are several thousand patients who carry the diagnosis of hypertrophic cardiomyopathy. And it'll be really interesting to understand what it is about those patients and their families that make some people have the disease and some people don't. That's the problem of rare variable penetrance. And then understanding how to classify variants of uncertain significance, which we're stumbling across a lot when we do genetic testing. So we really need to figure out what it is, use the electronic record to understand what phenotypes are associated with rare variants. And then I think this is sort of pie in the sky, but I've heard Nancy Cox say this, and I think I believe this. So if you have a complete picture of the genomic basis of disease, you then have a complete, and you have a sense of variability in the way that disease presents. You could, in theory, compute the genomic contribution, the social determinants of health contribution, the environmental contribution to variability in disease, and then understand that some diseases are 80% written in your genomes, and some diseases are 2% written in your genomes and 98% written in other omes. So I think that that's one of the real challenges and the real opportunities of a resource like this. And I'm sure there are many, many others. So I don't want to, those are my sort of, I call this musings because that's exactly what they are. And I think I'll just go right to the Q&A.
Wow. It makes me think of, I think the parable, if you want to go fast, go alone, but if you want to go far, go together. I think that's really fitting here. Just a reminder for the audience, feel free to drop your questions in the Q&A. We will ask them here to the panelists. If we don't get a chance to hit your question, we'll commit to dropping an email to you and following up. We've done that at all the other town halls as well. I think for the first question, I might fire it to Judd Schneider. It's at NashBio, he's their Chief Technology Officer. We learned, I think, a new unit today, bred in Wales. But for our investigators here, can you give a sense for the types of files that they might be able to utilize from this project, kind of the size of those files, whether RNA-seq will be available as a part of this project? I know we've got a lot of great big data labs that are represented here on the line.
Yeah, absolutely. And thank you for that question. Yeah, RNA sequencing, unfortunately, is not a part of this study. It is purely germline genome data. And, our partners at Illumina and the Dragon pipeline really have provided us with a wealth of different data types. So we obviously start with CRAMs, which are coming off the sequencer, basically are the reads. And then we have BAM files for each subject with the alignment. And then we've got GVCFs for each of the individual subjects, their variants in a single file. And then we've got a multi-sample VCF that's also compiled. Sizes for some of these files, CRAMs are obviously huge. I mean, they're 75, 100 gigabytes per individual. And as you go down, they get a little bit smaller. So BAMs are about 65 to 70 gigs. VCFs are, about 7.5 to 8 gigs per subject. The awesome thing too about working with Illumina is that we have this data normalized in Plink format too. So it is by individual chromosomes. And, we're looking at chromosome 1 for the 35,000 samples is around 260 gigs. So in order to do GWAS, you're going to have to break it up into multi-step. We routinely do this inside NashBio with our imputed data, and it's about the same size as what we usually use. But yeah, any other questions? Happy to answer, and thanks for the question.
Yeah, super helpful. Thanks, Judd. Peter, to you, I would suspect your department will be one of the big beneficiaries potentially of this sort of data for researchers that maybe already have invested in on-premise GPU clusters, et cetera, to analyze these types of data. How might someone be able to access these types of data in the future and bring them down for analysis here?
Yeah, great question. And I think the honest answer is I think we're going to be navigating that. I like the the citation level sort of measurement here that we're going to have to take into account, because some of the consideration is going to be about where is the best place to do the analysis in terms of compute capability and cost, and some of it is related to cost, the cost of data transfer. There's also considerations around just integrity of the data and the like, and accessibility. So I think for all of those reasons, we are more and more likely going to be trying as much as possible to take kind of a cloud first approach. But we recognize that that's not always going to be the right answer, depending on the kinds of analyses that need to be done. So we're certainly going to remain very open to the sort of case-by-case considerations for what really is required to do the research. But I think we are going to be very deliberate about ensuring that, if the right way to do the research and the most sort of cost-effective way is to actually enable these activities within the cloud environment, then we may be having a bit of iterative discussion with some investigators to say, we recognize in the past that might have been the best way is to pull it down to a local server, but today it might actually be better to do that work in the cloud. And I think we're going to learn a lot as we go.
I think that's a fantastic point. It's very different moving around a truck of bread than a truck of blue whales.
Right.
Let's see. We've got how about for Paul Harris? I think we've got Paul on the line. How will the availability of the Alliance for Genomic Discovery resource impact BioView, Vantage, and other kind of core resources that our investigators may be used to using?
Yeah, thanks for the question, Travis. I think the value proposition of having this whole genome sequencing data on 250,000 participants with paired phenotyping data that's in the synthetic derivative is really just incredible. I mean, for perspective, that's more sequencing data than we currently have in the national All of Us cohort, and we're seeing publications on a regular basis coming out of that resource. So I think going just reiterating, I think it has tremendous impact for BioView, has tremendous impact for our local research community, and the fun part is just going to be sort of putting it together in a way that as many researchers as possible can take advantage of it.
Fantastic. So as we've talked a lot about kind of the technical details, how about maybe Ellen Clayton, if you want to say a few words about, you've been at the center of navigating us through BioView and all of the ethical challenges and considerations around that resource. I can't imagine how many more ethical challenges might come up as we look at this AGD initiative. How do we face that as an organization, and what kind of guardrails do we already have in place? Ellen, if you're speaking, you may be on mute. I see you're highlighted, but. And if we need to, we might give Ellen a second of me to call you out there, Ellen. Maybe for Will while we're dealing with that AV. Will, do you want to say a few words about the ICA platform and some of the options that might be available to investigators if and when that becomes available to them?
Sure. Yeah. Thanks, Travis. So so ICA stands for Illumina Connected Analytics, as previously mentioned, and it's our data platform that sits on top of AWS. And it's really a modular system that's designed to accommodate users that are across the spectrum and their technical expertise. So someone that lives at the command line in a Jupyter notebook, Elizabeth Annie mentioned this before, but whether or not they want to run R, Python, what have you, they can go and they can deploy access libraries that are available. We also have a UI called cohorts that will be launched this quarter that will allow someone that's maybe not as, doesn't live at the command line, but would like to do cohort analysis, like to look at a set of individuals with a specific ICD-10 code, run a GWAS, run an analysis, but not from the command line perspective. So all of that is there. And there's also a data warehouse that's with Snowflake under the hood, so it does scale accordingly. And so there's, I think, some value to be had there for a lot of variation in user type. And lastly, it does abstract some of the complexity from the cloud, and that is the compute resources needed to be spun up, et cetera. It accommodates and deploys based on use and need of the compute lift that's for a specific query or analysis that's run. So it is there. It is the central kind of trusted research environment, as we call it, for the data and the structured clinical data and the VCFs and the genomic data, and it's available. And definitely we're happy to engage with those that need some guidance along the way. Thank you.
I think, Will, certainly, as Peter said as well, it's a new environment. I think that many of us are moving into with cloud first, and it's going to be a learning challenge, but it's certainly great to have a partnership like this as well. Dan, this might be the last question because I think we'll show the results of the poll that we opened up with, but thinking about our students and junior investigators that may have just learned about Mega and now are trying to decide whether to move their project forward on the Mega platform with BioView or moving to AGD, how would you kind of mentor them, and what kinds of questions can still be asked on the data that have already been run, and what kind of questions are better off waiting for one of the milestone deliverables that Elizabeth Ann shared with us on the timeline?
I think that really depends on the kind of question you're asking. I think that Nancy Cox and many investigators in VGI have shown us the real value of the array data. I mean, you can use the array data to construct predictors of transcriptional activity. You can use array data to construct predictors of circulating protein values. You can use array data to construct polygenic risk scores. You can use array data to understand to perform simple or not so simple GWAS. Those all focus on common variants, and I think the opportunity here is to understand the role of rare variants. They'll be rarer, so that you have in a population of 100 people, you might have one person that has an interesting variant, but they're high effect size. And then the other question is this business of variable penetrance, and how many people that really are there with Mendelian diseases that they don't know about but affect them or their families, and understanding why it is that some people have terrible phenotypes and what some people don't. So I think it just depends on the question you want to ask, but this gives us the opportunity to pose a lot more questions. I won't say one's more interesting than the other, but a lot more questions.
All right, I think with that, maybe we will put up, I think we'd be interested in seeing the results of the poll. Krista, if you want to share that. We opened with the question of what do you want to see? I think we had a clear winner with epigenomics. And then maybe Paul, if you have a second, maybe just one more. When will whole genome data from the first joint call set be available to our investigators for folks that want to have that standardization? Is that it was on? I think it was on Elizabeth Ann's timeline, but when can we expect for it to be fully available?
Yeah, I just wanted to make sure that that came through. That that should be coming into the Terra environment and available for researchers next month.
Fantastic.
So, thank you.
Super exciting. I mean, this is so fantastic. Will, special thanks to you for joining us here today. And Leland, the group together, this is such an amazing project. I think it continues to just push our enterprise and research enterprise especially forward. Thank you all for your participation in our town hall today. And with that, I think we will just mention that on the next town hall, augmented reality at VUMC. So this one's going to be really exciting. We've talked a lot about kind of the soft data side of things. We're going to talk more about hardware and some of the really interesting things going across the enterprise in this space. So really excited to host this in February. And so we've got a few others listed here. As always, feel free to go to the Office of Research site below, where you can catch recordings of any previous town halls, and also sign up to have these automatically added to your Outlook calendar so you don't miss any of the updates as they come across, because they've all just been exciting. So thank you all again. Have a wonderful rest of the day. Take care.